Hi everyone. In this video, we take a step further within the topics of molecules and compounds we learned in the last video, and we will learn about different macromolecules. In this video, particularly, we'll focus on carbohydrates and lipids. But before we get into more about these macromolecules, we want to know how these are actually formed. So from the mid-sized molecules, like what we've learned, um, these macromolecules are made of individual units called monomers, which are examples are like these, where they link together, basically polymerizing and form big polymers such as proteins and nucleic acids and carbohydrates, so on. And what macromolecules are is basically they are very big polymers that are made up of these tiny units called monomers linked together like this. So when we look at macromolecules that are polymers, those are like proteins, carbohydrates, and nucleic acids. But then lipids are not actually a polymer. They are a macromolecule, but not a polymer. So how do we actually assemble and break apart these polymers? This is through a process called polymerization, where they use energy inputs like ATP, and it literally joins together these individual units of the molecules. And this is non-spontaneous, which means that it requires energy input and that it won't happen on its own. Now, most times these monomers end up polymerizing through the process called condensation or dehydration reaction, which basically removes water molecules from the reaction. So when they get joined together, a water molecule forms and gets basically dehydrate that molecule. Hydrolysis, on the other hand, is the opposite reaction of condensation reactions, where polymers are broken apart by adding a water molecule into it. So the first macromolecule that we're going to talk about is carbohydrates. And this is one of my favorite ones, with all the bread and all the comfort food and stuff like that. So what do carbohydrates actually do? So these carbohydrates are components of larger macromolecules, such as like polysaccharides, glycoproteins, glycolipids, and parts of DNA and RNA, like our nucleic acids. Then it also can provide structural um, uh, materials like cellulose and like chitin and peptidoglycan. So that's what these, they would give more structure. And then with another part of it involves indicating the identity of the cell by having certain molecules on the surface itself, giving it a unique identity for the cells themselves. And then it also provides energy storage like glucose, starch, glycogen, all in that system itself. So these are some examples of what carbohydrates look like. Some can be linear like these, some are helical like these, and then some are branched like these two. And it is generally composed of carbons, hydrogens, and oxygens. and typically in this formula. So if you have like a with about n equaling 3 to 7, depending on how it goes. And what we see is that, like, for example, if we have like 6 carbons, we have 12 hydrogens and 6 oxygens. So, like, this is the chemical formula for glucose. And the monomers of carbohydrates are called monosaccharides, and the polymer version of carbohydrates are called polysaccharides. So now in a monomer itself, they can have different number of atoms, such as these, like glucose, which has 6. But then ribose, on the other hand, only has 5 carbons. Um, it can also vary in the location of the carbonyl group that we talked about last time, where it's a C double bond O, like this one here. It can vary depending on which carbon atom it's attached to. Like in this one, it's the first one. In this one, it's the second one. And then depending on how it is spatially oriented, it can have a different geometry of how it's actually set up, depending on the positioning of like different rings and stuff like that. And then the spatial arrangement of the hydroxyl groups can be different depending on how it's oriented and placed. And these are like structural isomers, which basically have the same chemical formula but different properties. So in this case, the example would be like glucose and galactose. They basically have the same chemical formula. However, it's the difference in the positioning of that hydroxyl group. Now with polysaccharides, what happens is that they have a long polymer of monosaccharides monomers. Those monomers join together by a bond called a glycosidic bond. And the simplest polysaccharides are disaccharides, which are basically 2 monosaccharides together. These monomers can be identical or different depending on the type of sugar it is. Like, for example, for sucrose, it's glucose and fructose, which becomes sucrose with by taking out a water. So like it's a dehydration reaction that involves a glycosidic bond. This is what you would call a glycosidic bond. Okay. Now, some common examples of monosaccharides and disaccharides include, so for monosaccharides, those are individual carbohydrate monomers. Some include glucose, fructose, galactose, and ribose. But then when we look at like disaccharides, which are basically covalently linked by a glycosidic bond, we have like maltose, we have sucrose, we have lactose. Those are just some examples. Now, there are 2 different types of glycosidic linkages in polysaccharides, and these kind of allow it to kind of understand what the function of these carbohydrates are. So when you have like an alpha 1,4 glycosidic linkage, that means it ends up with like energy storage polysaccharides. Whereas if you have like a beta 1,4 glycosidic linkage, those are structural polysaccharides. And what happens is that both of these have bonds between the first and fourth carbon hydroxyls, but how they're oriented is different. So like with this here, the alpha glycosidic linkages, what happens is that these are oriented in this manner, like this. But then when we have like a beta glycosidic linkage, they are in opposite directions, that it is oriented differently than this one, giving its the structure ends up determining the function of the bond, the molecule itself. So now energy storage polysaccharides, these are primarily linked by alpha glycosidic bonds. The polymers themselves can either be branched or unbranched, and some examples include like starch and glycogen. So starch is more where plants store sugar as starch, and then animals ends up storing sugar as glycogen. And these bonds are easily broken apart by digestive enzymes. With starch and glycogen, they can be broken down to release more glucose monomers when we need energy to produce a certain amount of ATP, the powerhouse of the cell, basically. Now, with structural polysaccharides, these are primarily joined by beta glycosidic linkages. These are basically unbranched, but when joined to other polymers, they end up forming like sturdy sheets or fibers. For example, in plants and algae cell walls, they end up giving structure to it, and the strands themselves are connected by hydrogen bonds. With fungi and exoskeletons of like insects and crustaceans, chitin is the structural polysaccharide. And these are usually modified using like acetyl amino groups, like these guys here. And they are basically the parallel strands end up being connected by hydrogen bonds like this. Now in bacterial cell walls, there's something called peptidoglycan. These are structural polysaccharides in the bacterial cell walls. And these are similar ways they are modified by amino acids, and the parallel strands are connected by peptide bonds, and that's usually with like proteins and all that, which we'll cover in a little bit. So these are your structural polysaccharides. Now we're going to branch off into lipids. And these lipids basically function as a couple of different functions, including energy storage, cushioning, and insulation, depending on where it's located. Major cell membrane component, like the phospholipid bilayer, and then precursors for certain vitamins, like vitamin D3 and sterols, such as cholesterol and testosterone. So what do lipids look like? Lipids are basically carbon-containing compounds found in organisms that, and they are largely nonpolar, because we can see how many carbons we have and not many polar functional groups. They are not exactly polymers, and they do not dissolve in water, because the major component of it is hydrocarbon based, and it's called a fatty acid. And the hydrocarbon chain bonded to a carboxyl group, the COOH functional group. So now these are non-polar, primarily because they only contain carbons and hydrogen. So what type of lipids are found in our cells? In our cells, we find three types, including triglycerides, phospholipids, and sterols. So some are more like this, where there's a glycerol molecule where it ends up with a fatty acid next to it. Some examples are like fats and oils. Then we have phospholipid, which is a phosphate head with fatty acid tails. And some of the examples of these are part of like the cell membranes and the organelles. And then there are some sterols, which are have ring-based structures, where some of them could be like cholesterol, steroid hormones like testosterone, estradiol, and cortisol. So the structure of fats itself usually contains um, uh, three fatty acids linked to glycerol. These are also called triacylglycerol or triglycerides. There are two types of fats where it's like saturated and unsaturated. The saturated fats consist of only single bonds between the carbons of the hydrocarbon chain. And then there are one or more double bonds in the hydrocarbon chain. And what we see is that the bond saturation in here, like whether depending on the amount of double bonds or single bonds that you see, it actually affects the physical state of the lipid itself. So if it's very saturated, what happens is that it gets compact and it becomes solid at room temp. But when it's unsaturated, where it's like more double bonds and stuff like that, what happens is it's like oil, where it's like fluid. It's not as compact as what you would see with saturated fats. So now the fluidity of the lipid depends on the length and saturation of these different hydrocarbons. So if we have like butter like this or like these are more saturated ones. Then there are saturated lipids with uh long hydrocarbon tails like these, and then unsaturated ones where they are like these oils and stuff like that. And when it comes to room temp, highly saturated ones are like basically butter and animal fat, where they are solid. And then vegetable oils and fish oils are more liquid at room temp. And then when we look at saturated lipids, what happens is that these with the extra long hydrocarbon tails, such as waxes, end up forming stiff solids like these beeswax over here. So the hydrogenation of fats is basically a chemical process that allows the unsaturated fats to be converted into saturated fats by breaking the C double bond carbon bonds and adding more hydrogens into the process. So if we put in more like hydrogens into this, it will become more solid. So liquid oils like vegetable oils can turn into fats that are solid at room temp. And this usually ends up increasing shelf life or texture for the baking. Many different things that you want to use, like a higher temperature in melting for like frying. And it's definitely cheaper than butter. However, it is very bad for your heart. So hydrogenated ones, saturated fats, don't go too crazy with them. You don't want to get yourself into like cardiovascular disease and all that. So next, we're going to talk more about the structure of steroids. Steroids are a family of sterile lipids with 4-ring structures. An example of them is like cholesterol, which is basically an important component of plasma membranes within many different organisms, and it can lead to the production of like a steroid hormones such as testosterone and uh, estradiol, and it can also help uh, form like vitamin D and bile salts. So when we look at the structure of membrane lipids, what happens is we have like a membrane steroid or a membrane phospholipid. Some of them are like in this hydrophobic area where they go in towards the membrane itself and then keep the polar hydrophilic sides outside. And it kind of looks like this, where it's space filling and all that. But then with membrane phospholipids, what happens is that there's a part of it that's polar, that's a phosphate group. And then we have a fatty acid kind of part of it where it stays within the hydrophobic side of it. These membrane lipids are amphipathic, and we will talk about those soon in the lecture in the second week. And so with phospholipids and water, what happens is that when it's placed in solution, the phospholipids form membranes that where the phospholipid heads, like these phosphate heads, end up interacting with the water, whereas the fatty acid tails end up facing the other set of uh, the hydrophobic tails. That way they don't get the have issues with having a hydrophobic tail next to water and having it all separate. So basically what happens with phospholipids, they can be like a micelle here, like this, or they can be a lipid bilayer where they are on top of each other. So like the tails, the hydrophobic tails want to be close to each other, avoiding any water that's on its surroundings. And phospholipids are major constituents of plasma membrane, where it looks like this. The phospholipids themselves face the water side, and then the uh, hydrophobic tails are basically um, gathered together here. And basically, they kind of contribute to the dynamic nature of the plasma membrane. And now we'll continue into the next video with nucleic acids and proteins.